So today we are going to discuss about the synapse. Synapse is the junction between two nerve cells with a minute gap, and this gap allows the transmission of impulse by neurotransmitter bridge. Now synapse is a junction or is a connection between the two neurons or the two nerve cells. Now for example, we saw the structure of nerve cell. This is the cell body. This is the axon which arises from the cell body. This is the nucleus. This is axon. It gets divided into terminals and the terminals have a swollen end. That swollen end is called as the axon bulb. Whereas from the cell body, projections they arise. These are the dendrites. These projections are called as dendrites. Fiber-like structures, those are called as dendrites. Now, here is another cell. This is the axon of another cell. This is nucleus. Axon ends into a terminals, and these projections, these are the dendrites of another axon. Now here, suppose we name this as axon 1, neuron 1 and neuron 2. Now here, the gap which is there in between the axon end bulb of one neuron and the dendrite of another neuron, the gap which is there in between the axon end bulb of one neuron and the dendrite of another neuron, this gap is called as the synapse. It is the junction between the two nerve cells or two neurons. The gap, it is very small gap in between axon end bulb of one neuron and in between the dendrite of another neuron. That small gap or that minute gap is called as synapse. Now the neuron carrying the impulse is called as presynaptic neuron. Now if the impulse is transferred from one neuron to another neuron, so suppose this neuron number 1 is transmitting the impulse to neuron number 2. So the neuron which is carrying the impulse, this first one, will be called as the presynaptic neuron. Whereas the neuron which is receiving the impulse, that will be called as postsynaptic neuron. So the neuron which carries the impulse is called as presynaptic neuron, and the neuron which receives the impulse is called as postsynaptic neuron. The gap between these two is called as the synaptic cleft. Now the gap which is there in between the axon end bulb of one neuron and the dendrite of another neuron, which we called as the synapse. In other words, it is called as synaptic cleft. So, here is the whole diagrammatic structure of the synapse. This is the axon end bulb of one neuron, and this is the dendrite of another neuron, and this gap which is there in between them, this gap is called as the synaptic cleft. Now here, the membrane of the axon end bulb, this membrane is called as presynaptic membrane, and the membrane of the dendrite is called as postsynaptic membrane. On the presynaptic membrane, there are channels or there are gates, voltage gates of calcium. There are voltage gated calcium channels which allow the neurotransmitters. Now what are neurotransmitters? The neurotransmitters are the substance which help to travel the impulse from one neuron to another neuron. And where are they situated? They are inside the axon end bulb. Inside the axon end bulb, there are circular vesicles, round vesicles present over here. And these vesicles, they have neurotransmitters in them. And about 2000 to 10,000 molecules of neurotransmitters are situated or are present in one vesicle, or each vesicle has about 2000 to 10,000 molecules of neurotransmitters. So there are vesicles in the axon end bulb, and these vesicles have neurotransmitters in them. Whereas the presynaptic membrane has channel gates. or voltage gated calcium channels which allow the neurotransmitters to pass through it into this synaptic cleft, into this synaptic gap. And on the dendrite membrane or the postsynaptic membrane, there are channels. These red ones are the potassium channels, these black ones are the sodium channels, and also there are chemoreceptors which are present on the postsynaptic membrane or the dendrite membrane. Dendrite membrane has two channels. One is for potassium ions and another is for sodium ions. Also there are chemoreceptors present on the postsynaptic membrane. So this is the structure of the synapse. Now the process by which the impulse from presynaptic neuron is conducted to the postsynaptic neuron is called as synaptic transmission. Now the process by which the impulse is conducted from one neuron, from presynaptic neuron to the postsynaptic neuron, that process is called as synaptic transmission, means transmission of the impulse, or transfer of the impulse. That is called as synaptic transmission. It is a one-way process which is carried by neurotransmission. Neurotransmitters, as we talked, these neurotransmitters, they help to pass on or to conduct the impulse from one neuron to another neuron. So this synaptic transmission is a one-way process. It is done in only one direction. It is not reversible. So it is one-way process and it is carried by neurotransmission. Depending upon the space, that is the synaptic cleft, and the way of conduction takes place. The synapse is divided into two types. Now depending upon the space, this synaptic cleft, depending upon this space and how the synaptic transmission is done, or how the synaptic transmission occurs, depending on these two things, the synapse is divided into two types. First is electrical synapse and the second is chemical synapse. Now, as we saw the structure of synapse, the axon end bulb of one neuron connects with the dendrite of another neuron through the synapse. The membrane of the axon end bulb is called as the presynaptic membrane, whereas the membrane of the dendrite is called as the postsynaptic membrane. Here on the presynaptic membrane there are voltage gated calcium channels, and on the membrane of the dendrite, that is on postsynaptic membrane, there are channels for calcium, sodium, and there are also receptors. These are called as chemoreceptors, or they are also called as neuroreceptors. Now here what happens, in the axon end bulb there are vesicles, circular vesicles, and these vesicles, they have neurotransmitters. For about each vesicle is having for about 2000 to 10,000 neurotransmitters in them. So these vesicles are called as synaptic vesicles, and they are having about 2000 to 10,000 molecules of neurotransmitters. Now here what happens, during a chemical synapse, the impulse reaches the axon end bulb. Now when the impulse reaches the axon end bulb, this impulse is transferred to the dendrite of another neuron. Now how it is transferred, that we are going to see. As this impulse reaches the axon end bulb, what happens? The calcium channels open and the calcium ions move into the axon end bulb. Now as this impulse reaches the axon end bulb, it triggers the calcium channels. It triggers the calcium channels and they open, and the calcium ions which are present here, over here, these calcium ions, they move inside the axon end bulb. These calcium ions, they move inside the axon end bulb. As the impulse reaches the axon end bulb, it triggers these calcium channels. These gates or these calcium channels, they open, and the calcium ions which are present over here in the synaptic cleft, these ions, these calcium ions, they move inside the axon end bulb through these calcium channels, through these voltage gated calcium channels. Now as these calcium ions they enter inside the axon, what happens? They bind to the presynaptic membrane. They bind themselves to the presynaptic membrane. As calcium ions they enter inside the axon and bulb, they bind to the inner presynaptic membrane. They bind to the inner side of the presynaptic membrane. Now this binding of the calcium ions to this presynaptic membrane triggers the synaptic vesicles to release the neurotransmitter. These synaptic vesicles release the neurotransmitter by exocytosis. Now here what happens? As these calcium ions they bind the inner side of the presynaptic membrane, this binding of the calcium ions triggers the vesicles. These synaptic vesicles are triggered by it, and as these vesicles they reach here near the presynaptic membrane, these vesicles they rupture themselves. These vesicles they get ruptured, they get broken down, and as they get broken down, the neurotransmitter which is present inside them, this neurotransmitter is released. These calcium ions they get binded with the presynaptic membrane, and this triggers the vesicles. These vesicles they get ruptured, they get broken down, and from these vesicles the neurotransmitter molecules which are present inside them, these neurotransmitter molecules are released. Now as these neurotransmitters as this neurotransmitter is released, the process by which this neurotransmitter is released, that process is called as exocytosis. The neurotransmitter then binds to the chemoreceptors or the neuroreceptors on the postsynaptic membrane, and a complex is formed. Now as this neurotransmitter molecules are released, these neurotransmitter molecules they are released into the synaptic cleft. They are released into the synaptic cleft, and as they are released, these neurotransmitter molecules what they do? They bind with the neuroreceptors. They bind with the chemoreceptors or the neuroreceptors which are present on the postsynaptic membrane. They bind together, and as they bind together, here a complex is formed. A complex of neurotransmitter molecules and the receptors. This complex is formed because of the binding of neurotransmitters to the receptors on the postsynaptic membrane. Now as this complex is formed, this complex opens the sodium channels on the postsynaptic membrane. Now as we know that on the postsynaptic membrane there are channels for the ions, potassium channels and sodium channels. Now as this complex is formed, this complex triggers or this complex opens the sodium channels. Sodium channels get opened because of this complex. Now what happens? As the sodium channel opens, the sodium ions present outside move in or enter through these channels, and the inner side becomes electropositive and the outer side becomes electronegative. As we talked about this, that suppose this is the membrane, this membrane. Now here inner side is electronegative and outer side is electropositive. This normal case is said to be polarized membrane. This is the polarized membrane. Now this membrane, we are talking about this membrane of the dendrite. This membrane outer side has positive charge, it is electropositive. Inner side has negative charge, means it is electronegative. Now as these sodium channels they open because of this complex, the sodium channels open, the sodium ions which are present outside the membrane, here the sodium ions which are present, because of these sodium ions there is positive charge outside on the outer side of the membrane. So these sodium ions they enter inside. as the gates open, as the channels open, the sodium ions, they enter inside the membrane. Now as these sodium ions, they enter inside the membrane, what happens? This electronegative charge becomes electropositive, inner side becomes electropositive and outer side becomes electronegative. We talked about this here. As the stimulus is given to the membrane, what happens? As the stimulus is provided to the membrane, at one point the sodium ions which are present in on the outer side of the membrane, these sodium ions, they enter inside and the inner membrane becomes electropositive and the outer membrane becomes electronegative. Same thing happens over here. Because of this complex, because of the complex of neurotransmitter and the receptors, because of this complex, the sodium channels, they open. As the sodium channels open, the sodium ions which are present outside the membrane, they enter inside the membrane. And as these ions, they enter inside the membrane, the inner side of the membrane becomes electropositive and the outer side of the membrane becomes electronegative. Now here the complex is formed. Here the complex is formed, or we said that the impulse has been generated. Here as the charges get exchanged, as the charges are exchanged, we say that the impulse has been generated over here. As the sodium ions enter inside the membrane, the sodium ions, they turn the electronegative charge of the membrane inside into electropositive and the outer side becomes electronegative. Here we say that the impulse has been generated. Now this impulse is conducted from dendrite to cyton and the axon end bulb of the another neuron, and from the axon end bulb to the dendrite of another neuron by synapse. Now as this impulse was transferred or transmitted from one axon end bulb of one neuron to the dendrite of another neuron. Now here we said that the impulse has been generated. This impulse is then transmitted from this dendrite, it is transmitted to the cyton. From the cyton or the cell body, it is transmitted to the axon. From the axon, it is transmitted to the axon end bulb of this neuron, and then from the axon end bulb of the neuron to the dendrite of another neuron in the same way, with the help of the synapse. So as we suppose this is the structure of the neuron. If we say this is the structure of neuron, these are the axon terminals, this is the axon end bulb, and these are the dendrites. Now if the impulse is traveling from this axon, if the impulse is traveling from this neuron to another neuron, suppose this is another neuron, these are the dendrites. Now here this impulse have been transmitted to the axon end bulb. From axon end bulb it will be transmitted to the dendrites of another neuron, and from the neuron it will be transmitted to the cyton, this cell body cyton. From the cyton it will be transmitted to the axon. From the axon again to the axon terminals, and from this axon end bulb again to the another neuron. So in this way the impulse is traveling from one neuron to another neuron through the synapse. Now here, if the neurotransmitters, after this impulse is generated and it is passed on to the cyton by the dendrite, after that the neurotransmitter is still here in the synaptic cleft. Now if this neurotransmitter remains in the synaptic cleft, it will keep on binding with the receptors which are present on the postsynaptic membrane, and it will keep on generating the impulse again and again. So this neurotransmitter should be removed or it should be broken. Otherwise what will happen? If the neurotransmitter remains here in the synaptic cleft, it will keep on binding with the receptors which are present on the postsynaptic membrane, and it will keep on generating impulses. It will keep on making the complex. This complex will again open the sodium gates, and again the sodium ions will enter inside. So again the another impulse will be generated. So this neurotransmitter, it should be removed or it should be broken over here. So because for this an enzyme is secreted by the postsynaptic membrane. Now for breaking down this neurotransmitter, this postsynaptic membrane secretes an enzyme. For example, if the neurotransmitter is acetylcholine, then the postsynaptic membrane releases an enzyme. That enzyme is called as acetylcholine esterase. If this neurotransmitter is acetylcholine, the dendrite membrane, the postsynaptic membrane releases an enzyme that is called as acetylcholine esterase. Now, if this acetylcholine should be broken down, and that's why the acetylcholine esterase is secreted by the postsynaptic membrane. Suppose this is acetylcholine enzyme. This acetylcholine should be broken down. For that, this postsynaptic membrane secretes an enzyme that is called as acetylcholine esterase. This acetylcholine esterase, what it does, it breaks down the acetylcholine into two substances. So here, this acetylcholine is broken down into two substances. One is the acetic acid, and the other substance is choline. So this acetylcholine is broken down into two substances by the acetylcholine esterase enzyme. First substance is acetic acid and the second substance is choline. If these two substances which in which the acetylcholine is broken down, that is acetic acid and choline. Now what these two substances do? These two substances, they again enter the axon end bulb. They enter inside the axon end bulb and they help in the formation of the synaptic vesicles. They help to generate the new synaptic vesicles. In this way, this acetylcholine is divided into two parts or two substances, acetic acid and choline. These acetic acid and choline, they again enter the axon end bulb and they help in the generation or in the formation of the synaptic vesicles. Now if the neurotransmitter is adrenaline, then the enzyme which is required to break down this adrenaline is monoamine oxidase. If the neurotransmitter is acetylcholine, the enzyme which is required to break it down will be the acetylcholine esterase. And if the enzyme is adrenaline, if the neurotransmitter is adrenaline, the enzyme which is required to break it down will be monoamine oxidase. Now this process is one-way process. Why it is a one-way process? Because here on the axon end bulb, also on the on the presynaptic membrane, here are the channel gates. Calcium channel gates are present on the presynaptic membrane. As we said that the axon end bulb has the neurotransmitters. It has the vesicles which contain the neurotransmitters, and these neurotransmitters are present only in the axon end bulb. They are not present in the dendrite. So, and on the postsynaptic membrane there are channels: calcium channel, potassium channel, sodium channel, and the receptors. Here these are the receptors and the channels which are ion channels which are present on the dendrite membrane. these receptors are not present on the presynaptic membrane. They are only present on the postsynaptic membrane. So here these receptors they receive the neurotransmitter on the dendrite membrane, on the postsynaptic membrane. These are present only on the postsynaptic membrane. They are not present on the presynaptic membrane. Whereas the neurotransmitters are only present in the axon. They are axon end bulb. They are present in the synaptic vesicles, in the axon end bulb. They are not present in the dendrite. So the impulse which is generated, the direction of the impulse which is conducted, or the direction of the impulse which is conducted, it is from the axon end bulb to the dendrite. From the axon end bulb of one neuron to the dendrite of another neuron. This cannot be reversed because there are no receivers on the axon membrane. There are no receivers on the presynaptic membrane. The receivers are only on the postsynaptic membrane. Whereas there is no neurotransmitter in the dendrite. It is only in the axon end bulb. That's why the conduction of the impulse is done from the axon end bulb to the dendrite. From the axon end bulb of one neuron to the dendrite of another neuron. It cannot be reversed from the dendrite to the axon end bulb. So that's why it is a one-way process. And now we will compare the chemical synapse with the electrical synapse. Now in the chemical synapse that we saw, the synaptic cleft which is there between the axon end bulb and the dendrite of the other neuron, that synaptic cleft is about 10 to 20 nanometers. Means it is wider. The synaptic cleft which is there in between the axon end bulb of one neuron and the dendrite of another neuron is about 10 to 20 nanometers. Whereas in electrical synapse, the synaptic cleft which is there is about 0.2 nanometers. Means this is narrower as compared to the chemical synapse. The synaptic cleft in the chemical synapse is wider than the synaptic cleft in the electrical synapse. Second thing, the transfer or the conduction of the impulse is done by neurotransmitters. As we saw in chemical synapse, that the neurotransmitter, a chemical is secreted in the released in the synaptic cleft by these synaptic vesicles. This chemical is the neurotransmitter, and the impulse is transmitted from the axon end bulb of one neuron to the dendrite of another neuron with the help of this neurotransmitter, with the help of this chemical. Whereas in the electrical synapse, the conduction is done by transfer of the charge or potential from one node of Ranvier to another node of Ranvier. For example, if we draw here, suppose this is the cyton, this is the cell body of the neuron, this is the axon, and these are the axon terminals. Here are the dendrites which arise from the cyton. Now, and this is the nucleus. In the beginning, as we saw the structure of the neuron cells. Now this is the structure of neuron. Now in electrical synapse, what happens? This the conduction or the transfer of the impulse is done by the charge or the potential. Now suppose here are positive charge, inside is negative charge, outer side of the membrane is positively charged, whereas the inner side is negatively charged. These charges, what happens? These charges are transferred from one node of Ranvier to another node of Ranvier. These charges, they jump from one node of Ranvier to other node of Ranvier. Here also, these jump from one node of Ranvier to other node of Ranvier, and from here to the axon end bulb, and from axon end bulb to the dendrites of the another neuron. In this way, the conduction of impulse is done in case of electrical synapse. Whereas in case of chemical synapse, the conduction is done through the chemical called as neurotransmitter. Here, no chemical is used. That's why it is called as electrical synapse. The charges are responsible for the transfer of impulse. That's why it is called as electrical synapse. Here the chemical is responsible for the conduction of impulse. That's why it is called as chemical synapse. Now next, that is synaptic vesicles with neurotransmitters are present. Now here in the axon end bulb, the synaptic vesicles are present, which have the molecules of neurotransmitter inside them. Synaptic vesicles are present in chemical synapse, whereas in the electrical synapse, the synaptic vesicles are absent. Here no chemical is required for the conduction of the impulse. That's why synaptic vesicles are also absent over here in the electrical synapse. Mitochondria numerous is present in the axon end bulb. Now to carry on this process, all the processes in chemical synapse, from secretion of, from rupturing of these synaptic vessels, secretion of the neurotransmitter, receiving of the generation of the impulse, for everything energy is required, ATP is required. So that's why here mitochondria numerous are present in the axon end bulb. More in number, the mitochondria numerous are more in number in the axon end bulb in case of chemical synapse. Whereas here in electrical synapse, only few mitochondria are present in the axon end bulb. Postsynaptic membrane has chemoreceptors. As we saw, this postsynaptic membrane, it has chemoreceptors which receives the neurotransmitters. These neurotransmitters molecules, they get binded on the postsynaptic membrane to the receptors, chemoreceptors or neuroreceptors, which are present on the postsynaptic membrane. Whereas here in electrical synapse, there are no chemoreceptors. No chemical is required for the transmission or conduction of impulse. So no chemoreceptors are present over here on the postsynaptic membrane. Now here lag period is about 0.5 milliseconds, and the speed of conduction of the impulse is slower in the chemical synapse. Whereas in electrical synapse, no lag is there, means the induction of impulse is faster as compared to the chemical synapse. Now here in the chemical synapse, this chemical is released. That chemical gets bind on the postsynaptic membrane on the receptors, and then these receptors, they open the channel gates for the sodium ions, and then the sodium ions which are present outside the membrane, they enter inside the membrane, and then the charges are exchanged. This process is same process is followed on the other neuron, on the, as this impulse is traveled from this dendrite to the cyton, from this cyton to the axon, and from axon to the axon end bulb of this neuron. From here it will be transferred to the another neuron. So in every junction of the axon end bulb and the dendrite, every junction has to go through this process, transmission or releasing of neurotransmitter, and then this neurotransmitter will act or bind with the complexes, bind and form the complex on the postsynaptic membrane. So this takes a lot of time. That's why the the conduction of the impulse is slower here in the chemical synapse. Whereas in electrical synapse, no chemical is required to carry on the conduction. The electric charges, they can jump from one node of Ranvier to another node of Ranvier. From first node of Ranvier to second node of Ranvier, from second node of Ranvier to third node of Ranvier. In this way, the electrical charges, they can jump from one node to another node. So that's why no chemical is required over here for conduction of the impulse. So that's why this conduction of the impulse in the electrical synapse is faster as compared to the chemical synapse. The next thing that we are going to see is the speed of the nerve fibers, the speed of conduction of impulse of the nerve fibers. The faster the speed of the conduction of the impulse of the nerve fibers is faster on the myelinated nerve. Now this we talked about, this is the myelinated sheath. So in the if the neurons are if the axons of the neuron is myelinated, or if the nerve fibers are myelinated, the speed of the conduction of impulse will be faster, 20 times faster as compared to the non-myelinated. So the myelinated nerve fibers, they conduct the impulse 20 times faster as compared to the non-myelinated nerve fibers. The thickness and the thickness or the diameter of the fiber. Now the thicker the nerve fibers, they show the faster conduction. Now if the nerve fibers are thicker, they show the faster conduction, means if they are thicker, the conduction of the nerve the conduction of the impulse will be faster. This kind of conduction is found in the squid and the earthworms. They have the thicker nerve fibers, so the conduction of the impulse is faster in them. There are few non-myelinated fibers which are present in the body of humans. That is C fiber, which is present in the skin. These are non-myelinated fibers and they are associated with the conduction of pain. The temperature and pH also affect the rate of impulse conduction. So along with this, myelinated and non-myelinated and the thickness and diameter of the nerve fibers, along with these factors, the temperature and the pH are also two factors which affect the conduction or the speed of the impulse conduction of the nerve.